Okay, so the third macromolecule that we focus on is proteins. And proteins are my favorite macromolecule because of their importance for life. Life has all of these different properties and these different functions that have to happen in order for something to be considered living. Proteins are the driving forces behind doing all of those functions. They are the little machines that do everything that is required for life to be considered life. And because of that, they have so many different shapes and orders and things like that. And if you remember, one of the fundamental, I said three fundamental things for understanding basically all of science, specifically biology. The first one, likes dissolve likes, opposites attract, and the third one, structure determines function. Well, proteins have so many different varieties in structure that determine their function. And if you mess up that structure, it ends up causing lots of problems and imbalances in homeostasis in the body. So here's an example, or here's showing you a lot of the different things that they do. They're enzymes. They're structural. They provide support within the cell and outside of the cell. There's proteins that store amino acids and things like that, like albumin in the protein of egg whites. And you have um, casein, which is a milk protein, which is made amino acids for baby animals. So you have that storage component. There's transport proteins that allow things to get across the cell membrane, which is so pivotal. It's how cells maintain homeostasis. Hormones, how cells communicate with each other. Receptors, how cells receive those hormones and transmit that information into the cell. Movement, contractile and motor proteins like cilia and flagella, actin and myosin, super cool proteins that allow cells to move. And also antibodies, which allow us to protect our bodies from infection. So they do all of these different things. And each one of them is very unique due to the structure of it. So in order to determine a protein, you basically have to know first that it's made up of these things called amino acids. That's the monomer of a protein, amino acids, okay? And every amino acid has the exact same repeating structure with one exception. Every single one has an amino group, this NH2 group, then a carbon attached to a hydrogen here with this R group, which I'll talk about in a second, and then an acid, a carboxylic acid group on this side. So we look for this repeating over and over and over again. The only place an amino acid differs is at this R side chain. The R means it can kind of be anything, essentially, though there are only 20 different types that exist or that we care about in biology. So what you look for is CHONs. CHON being the main part. So if you look at here, carbon, hydrogen, oxygen, and nitrogen. And the sulfur part is usually in R groups, but it's not in every single R group. That's why I have it in a parentheses. So remember, proteins are the only macromolecule that will have sulfur. That'll be important, especially when you look at some of the AP questions that we're going to look at later, or ones that'll be on your test. So here's showing you amino acids and their R groups. The R groups are highlighted in white, so they each have their own name: glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, proline. Okay. Now if you look, their basic structure is all exactly the same at the bottom, but we differ in these spots right here. In college, if you go into biochemistry, you have to memorize every single one of the 20 based on their R groups. I will not make you do that. But what we are going to focus on is what makes them different, what makes them polar or nonpolar. So remember, what makes something nonpolar? For an amino acid, it only has to do with the R group. Because the amino acid itself does have polar components to it, right? This oxygen draws the density over here. And there's probably less electrons over in this direction. But the R group itself, is it made of carbons and hydrogens only? Then it's considered nonpolar. This sulfur this doesn't actually affect it too much, so this still is nonpolar. These are all carbons and hydrogens. This nitrogen, there's not enough electron density draw, so it's still considered to be nonpolar. This is nonpolar, okay? So as you can see, nonpolar amino acids, their R groups tend to be nonpolar. And what do you think is true about, would they like to be near water or would they like to be further away? I want you to think about it. Because they're nonpolar, they're hydrophobic, and thus they want to be away from water. And that'll become important when we talk about how proteins fold and determine the shapes that they are. So amino acid, they're also polar ones too, as well. And those would be hydrophilic, and those would probably want to be around water. Okay? So amino acids are bonded through something called the peptide bond, which is essentially how the amino acid bonds through a dehydration synthesis reaction, forming a polypeptide, or adding an amino acid to the next amino acid, basically. So you can see here, in purple, we have the basic structure of the amino acid. Your peptide bond's always going to be this between the carboxylic acid carbon here with the acid group right there and the neighboring amino group. So you get rid of the OH from the carboxylic acid and the H, you form water, and then we form a peptide bond, which is a covalent bond between the amino acids. This is a very strong bond. Notice here we have some R groups. This R group would be more polar. Why? Because the OH draws electron density towards it. Same thing with this one. OH draws electron density towards this part of the molecule. It makes it a little bit more polar. Okay? So once you put a bunch of these things together, now this is just a different representation. This would be, the amino acid would be here with the R group. They're just drawing like a circle, almost like this is a string of beads. But this would just be each amino acid next to each other. And this is a polypeptide, a long chain of amino acids. And the way it folds is determined by characteristics that exist in these amino acids. Each one's unique, and they like to bond and interact with other ones in different ways. So the order of those amino acids, because of that fact, and the different R groups that are on those amino acids determine the structure of the protein. So the order of the amino acids determines the structure, or as we call it, the conformation, how the protein folds on itself to inevitably give it the structure it needs to do its job. Okay? So the structure will determine the function. Now there's 4 levels of protein structure that we have to look at. Every protein pretty much has all of these, with the exception some don't have the last one, which we'll talk about. But they all have these different levels. Primary structure is just the order of the amino acids, going in order, just saying what they are in order. So it's hard to really see here, but we have glycine, proline, threonine. I think that's glycine, threonine, glycine, glutamine, and saying that order. So if we want the primary structure, it's just what is the order of the amino acids in that polypeptide. Okay? Secondary structure is then how do those amino acids start to fold into these smaller structures within an overall protein called alpha helices and beta sheets. This is due to hydrogen bonding between adjacent amino acids and has nothing to do with the R groups at this point yet. So the R groups don't interact for this specific conformation. It's just how they like to fold into either a beta pleated sheet, which looks like this, and we can see the hydrogen bonding as these dotted lines, or the alpha helix, which shows dotted lines over here. A helix looks like a helix, which is basically like a spring almost, where a beta sheet looks like a, trying to think of how to, like the side of an accordion. That's what it reminds me of. I don't know. But okay, so that's secondary structure. How does it start to fold into these structures due to hydrogen bonding, either into alpha helix or a beta sheet? Then, if you look here, this is tertiary structure. It's how we put together all of the alpha helices and beta sheets in a single polypeptide together. Now, this folding is due to interactions between R groups. So you can see here, like these are both hydrophobic amino acids. They are going to want to be oriented on the inside of the protein because they want to avoid the watery environment on the outside. The more ionic and polar ones will probably want to bind more towards the outside, or the ionic, the negatively charged ones will connect to the positively charged ones because opposites attract. So remember, all important things in life boil down to hydrophobic, hydrophilic, likes dissolve likes, right? And opposites attract, and that's true within proteins especially. You can see hydrogen bonding. So all of these, the order of the amino acids determines how the protein folds because of these interactions between different R groups with different characteristics on individual amino acids. Quaternary structure then, we'll look at the overall protein structure when we have multiple subunits or multiple polypeptides that are put together to do an individual function. Some proteins only have tertiary structure. They do not have multiple subunits that come together. But if they do, that next level is called quaternary structure. A good example of that is actually hemoglobin, which is the structure you see right here. There's 4 different subunits, 2 alpha and 2 beta chains put together, and collectively they help to carry oxygen in our red blood cells. If they did not come together, they would not properly carry oxygen. So here's looking at all of them. Remember, primary structure is just the order of the amino acids of a protein. Secondary structure, how does it start to fold together into alpha helices and beta sheets with hydrogen bonding between adjacent amino acids? Tertiary structure is looking at How do all those alpha helices and beta sheets in an individual polypeptide come together to form an individual subunit? Remember, some proteins stop there, but if you have multiple subunits that all come together to do a function, that's called quaternary structure. A great example of how a simple change in primary structure can result in a change in secondary, tertiary, and quaternary structure would be sickle cell anemia. So sickle cell disease actually results from a single amino acid substitution. So we change one amino acid in the gene of an individual, or I should say nucleic acid, which eventually changes it into an amino acid, which we'll get into later in the year. And as a result, having one different amino acid changes the whole shape of the protein, causes it to no longer function the way it's supposed to, and as a result, causes a disease in people. So here's normal hemoglobin right here. Valine, histidine, leucine, threonine, proline, glutamine, glutamine. Notice we changed the glutamine to a valine. That's the one change that we make. That's it in the whole thing. And there's thousands and thousands of these amino acids. We change one of them. The difference between glutamine and valine, and you don't know this yet, you don't have to memorize this, but valine is a very nonpolar amino acid and glutamine is a polar amino acid. So it causes the whole shape of the protein to change just because we changed from a polar to a nonpolar amino acid. And it causes this hydrophobic region to be exposed when it normally wouldn't. And as a result, it causes different interactions between the individual subunits in the quaternary structure, and it forms these long chains as opposed to these separate individual proteins. And as a result, instead of normal hemoglobin and normal red blood cells, you get these sickling red blood cells in low oxygen environments, which causes pain and eventually will actually kill you. So it kind of shows you how you mess with that one little thing and you change one amino acid. You can see how, because it has different characteristics, it changes the shape of the protein and as a result causes a change in the function of the protein and thus a disease. So what determines protein conformation? So as previously mentioned, the sequence of amino acids is very important. And it also depends on the environment it's in. So if it's in a very watery environment, a protein may fold differently than if it's more in a hydrophobic type of environment. Or also, sometimes if it's in an acidic or basic environment, it changes the shape of it too. Or if it's in a really hot environment versus a cold environment in other organisms, because we have ours pretty much leveled out at 37 degrees Celsius. So what can sometimes happen, though, so a protein has a normal conformation, but if we mess with that somehow, either because we heat it up or we throw it an acid or a base where it's not supposed to be, it will mess with the interactions between the amino acids, change its shape, and thus change the function. That process is called denaturation. It's basically the unfolding of a protein. When you denature a protein, you do not affect the primary structure. The primary structure stays intact, the order of the amino acids. But you do mess with first the quaternary structure and tertiary structures, and then eventually the secondary structures if it's bad enough. And then you never will really mess with the primary structure unless you literally combust it, which would never happen in natural conditions. So remember, denaturation is just the unfolding or unraveling of a protein where it loses the structure that it needs to function properly. So what causes this? I mentioned these already. High temperature. If you imagine if you have a protein and it's folded, and it's supposed to, and intermolecular forces are holding it together. If you increase the temperature, it causes the molecules to vibrate faster, eventually disrupting the intermolecular forces, causing it to fall apart. So it'll no longer work. The shape changed. pH, adding hydrogen ions or OH negative ions to things will cause it to stick to different charged and polar and charged groups, causing it to misfold if it's in the improper environment. Like, for example, some things do function better in acidic places. Those proteins are meant to be in acidic environments, and they fold when they're in those environments properly. If they are in a basic environment or a neutral environment, they won't fold properly. Okay? Salinity or saltiness, because it's charged ions, can also bind to R groups, causing misfolding. So there's lots of different things that can cause proteins to denature. And what happens is after proteins are made, when we get into that later in the year, they're going to fold and spontaneously start folding, and it sometimes takes multiple steps before it finally forms its stable functional conformation. Other times, it needs help. There are special proteins called chaperone proteins or chaperonins that are protein folding proteins. They actually have a cap out. This is what it actually looks like. The cap opens up. The polypeptide will go in there. It provides a different microenvironment than that would be in the cell normally, and it helps it to fold and orient properly. So you can imagine if you have a mutation in the gene that codes for this chaperone protein, what would happen to the ability to fold proteins properly? It probably would be hindered or affected in a negative way, and therefore you would not be able to make a lot of proteins. There's some cancers that have issues with their chaperone proteins, actually. So super cool. We're going to have a whole unit on enzymes, but enzymes are a special type of protein that drives chemical reactions. And there's millions of different types of chemical reactions that happen in the body. Enzymes are a catalyst that helps to speed it up. And essentially the way they work is they help to grab the thing we want to react and put it in an orientation that makes it easier to react. But we're going to get into that a little bit more when we get into enzymes, which is the next unit that we're going to cover. Okay. So here's some quick questions. If you're studying cellular enzyme involving and breaking down amino acids for energy, where would you predict to find the amino acids in the part of the enzyme that will interact with fatty acids? Think back. What did I say we can break everything down into? Opposites attract, likes dissolve likes. Fatty acids are what? They're hydrophobic. So where would you find the amino acids that would interact with fatty acids? Probably on the inside, the hydrophobic region of the enzyme. Does that make sense? Because likes want to be around likes. What happens when a protein denatures? It has all of these things. Does it lose its primary structure? No, it does not. It keeps that intact. It'll likely lose its secondary and tertiary structure? Yes. Does it become irreversibly insoluble and precipitate? Like, does sometimes it do that? It can sometimes. Sometimes it can reversibly turn back into a functional protein, but usually it does not. It hydrolyzes into its component amino acid. It does not do that, right? It does not lose its primary structure. It's the same thing as number one. Its hydrogen bonds, ionic bonds, and peptide bonds are disrupted. Its hydrogen bonds and ionic bonds could be, but its peptide bonds would not because they're usually strong unless you literally combust the thing. Okay, so quick review of proteins. Remember CHON and sulfur because it's the only macromolecule that had sulfur in it of the four we're going to go over. Its monomer is amino acid, which will form peptide bonds to create polypeptides. Remember the basic structure of an amino acid with that amino group, the acid group, and the R group off of it. There's 4 levels of protein structure: primary, secondary, tertiary, quaternary. Primary is the order of the amino acids. Secondary, how it starts to fold via hydrogen bonding into alpha helices and beta sheets. Tertiary, all the alpha helices and beta sheets interacting together due to R group interactions between the amino acids, folding into a 3D structure. And quaternary, having multiple subunits coming together, interacting within a protein. Denature is the falling apart of a protein due to lots of different factors, changing its conformation. It's caused by high temperatures, varying pH, or salinity are the 3 main ones. There are other things too, but those are the 3 main ones. And proteins need to fold properly, and they fold due to their amino acid sequence and environment. Sometimes they need help via chaperone proteins. And lastly, enzymes are protein that help to drive chemical reactions and are biological catalysts. All right, that's it on proteins.